Hello, everybody, and welcome back to Drug Repurposing for Rare Diseases 2021. Really excited about this session today and really glad you're here to join us. So this session is all about the AKU story and the journey that the AKU Society and all their different collaborators has been on to take a drug from essentially inception right the way through to now being marketed and available and delivered to patients on a regular basis, at least in the UK and hopefully much wider soon. Before we dive into that, once more, it's worthwhile me just highlighting our event sponsors and thanking them all for making the event possible today. We're really enjoying how it's going and great to the interaction we got in the last session, so thank you all for that. With that, I'm going to stop my share and you can see my face and hopefully the faces of all the rest of my panel. So without further ado, I'm going to ask each of my panelists to introduce themselves, give you a little bit of background on how they tie into this wonderful story we're going to be diving into in depth over the next hour and a half. So first and foremost, Nick, please say hi.
Hi, yes, so I'm Nick. I am the chair and CEO of the AKU Society. We are a patient group that works on the ultra-rare disease alkaptonuria, or AKU, and I'm also the chair and co-founder of Find a Cure. Thank you.
Thanks a lot, Nick. Ranga, you next, please.
Hi, I'm Ranganath. Short version of that is Ranga. I'm a consultant at the Royal Liverpool University Hospital. I'm also the clinical director of the National Alkaptonuria Centre, and I've also had the privilege of leading the Develop a Cure clinical trials. So that's me.
Thank you, Ranga. Kieran, you're next on my screen.
Good afternoon. Yeah, I'm Kieran Scott. So I'm the head of projects and comms at the AKU Society. So I've been there for about 6 years. So I joined halfway through the whole Develop a Cure process.
Thank you, Kieran. Tony E, if you want to say hi.
Hi, I'm Tony Esmond, and I'm an AKU patient. And I also took part in the clinical study. I'm also a member of the trust board for the AKU Society.
Thank you, Tony. Krista.
Hi, I'm Krista Van Ken. I've worked in clinical research my whole career and specifically in rare diseases during the last decade. And I'm currently working as a consultant, but during the Develop a Cure project, I was acting as the clinical project manager for the clinical trials while working at a contract research organization that was focusing on orphan drug development.
Thank you, Krista. That's great. Jim, be great to hear your introduction.
Hi, I'm Jim Gallagher. I'm a professor at the University of Liverpool, and my research career has all been on cartilage and bone. And about 15 years ago, I started working on AKU, alkaptonuria, thanks to a request from Ranga. And I've been involved in a few ways. One is trying to discover how AKU leads to joint destruction. We also developed some models, and the mouse model of AKU is very important in today's story. And also, we've developed some methods to measure ochronosis, which is the one of the pathological features of AKU, not just in models, but also in people, for example, in the Develop a Cure trials.
Thank you, Jim. That's brilliant. And finally, Tony Hall.
Hi, I'm Tony Hall. So I trained originally as a physician and joined the pharmaceutical industry quite early on in my career. So my career has been spent mostly in drug development, and the last 10 or 11 years of that has been focusing solely on rare diseases. I got involved with Develop a Cure quite near the beginning in 2010 when we were just putting together the consortium. And I'm also the co-founder and trustee of Find a Cure. And my full-time job at the moment is I'm Chief Medical Officer at Helix.
That's brilliant. Thank you guys so much. So everyone, as you can see, we've got a really diverse panel, a rich panel, lots of different voices that are going to dive deep into this story and take us through it today. We are keen to hear questions from you and to try and answer them throughout this session. And we've got a good chunk of time to do it in. But there is a big story here, so do bear with us if we take a little time to get to those responses. My general aim is that we're going to have a little bit of an introduction to the story itself and the people who know it best, which you'll be pleased to hear is not me. And then from there, we're going to pick out a few key themes, which we're going to walk through with the panel here. throughout there. If we see questions that fit into that narrative, we're going to pull those out and hopefully have a good chunk of time at the end to really address the questions that you are submitting. So please put those questions in nice and early, get them submitted, and we'll do what we can to work through them during this session. But I'm sure all the people here will be glad to talk to you separately and be contacted through the platform or independently. So with that, I'm going to hand over to Nick and Ranga to take us through the early stage, the basic overview of what is this history of AKU, what is the story that you've got to share.
Great. Yeah, thank you very much, Rick. So I'm going to speak for a few minutes about the first few years of the AKU Society till around 2010, which was a key landmark for us. And then Ranga is going to go talk from 2010 till 2021. Very briefly, I'll tell you about AKU, but Ranga will go into more detail. AKU is an ultra rare disease, affects roughly 1 in 250,000 to 1 in 1,000,000. So very much on the ultra rare side of the spectrum. Both my boys, who are now aged 20 and 17, were diagnosed shortly after birth with AKU, and it's also called black bone disease because basically the bones and cartilage go black, so it's very severely debilitating as life progresses. And it was first identified, or first kind of really talked about, in 1901 by a doctor called Sir Archibald Garrod in London, who identified it as an inborn error of metabolism, which was the first time any disease, I believe, was actually shown to be inherited. So very much in all the medical history books. But yet 100 years later, by 2002, there was no patient group for it. And I think the real creation of this patient group comes down to a patient who sadly is no longer with us anymore, called Robert Gregory from Liverpool, and who went to the Royal Liverpool University Hospital and met there with Ranga, who was a doctor at the time at the Royal, or still is, and talked to him about setting up a patient group to really try and resolve this problem of the fact that AKU didn't have any treatment and was still completely, well, not very well known, and we didn't know many patients with it and all that. And so really from 2002 till today, that's 19 years, that is really the history, I believe, of the AKU journey. And then in 2003, Bob and Ranga set up the AKU Society. I was invited at the time because I contacted Bob about my children to join the Board of Trustees. And so we were founded officially in June 2003, okay? And Bob was absolutely instrumental in the first few years till he got very ill and sadly passed away. And without him, we would be nowhere near what we're doing now. There probably wouldn't be an AKU Society. And so really what we did is focused on what Ranga really seemed to have a plan for research. I'm not a scientist by background. My PhD was in social psychology. And until 2010, I was working in the international NGO sector doing solar power in Africa and doing AKU in my spare time. So was really reliant on what Ranga and then Jim were saying to actually develop a strategy. And as you can see here, with all these different dates that you see in there, it really is, I think, a kind of a model for how to do research into a rare genetic disease, stage by stage, and how each stage builds on the next stage. Obviously, I believe that if we'd had much more funds at the beginning, we'd have probably been able to go faster through it, but we still had to go through all these stages. So in 2005, one of our patients passed away and donated her body to science. And Ranga and a physician called Tim Helliwell then did the post-mortem, and this was published. It was the first ever publication, I believe, of an autopsy of a patient, and revealed some very interesting discoveries really about AKU and the pigment within the body. In 2008, we had a huge disappointment. There was a drug called nitisinone, which Ranga will explain a bit more about, which was a very promising treatment for AKU. And they were testing it in the US at the National Institutes of Health in a clinical study. And that study failed. And we know now it wasn't enough patients, didn't last long enough. The endpoint wasn't really sensitive enough. But that was really quite a blow to the AKU community. In the meantime, we were driving ahead. So as Jim said, he joined 15 years ago in 2006 when Ranga came to see him, and we funded a PhD someone called Adam Taylor, who was absolutely fantastic, and he developed a cell model of AKU. And then after that, we got some funding from the lottery, £500,000. The lottery has been a really good supporter of our initiative to do an animal model, which was difficult at first, but it ended up being a really good animal model of the disease, a mouse model of AKU. So a few other things. One is, I think, what was really important was the natural history study, which we did with some funding from a foundation called the Chillick Trust, which really allowed Ranga to develop the AKU Severity Score Index that was used as one of the endpoints for our following trials. And then I'll always remember, so the results of the NIH trial were presented to us at a meeting we organized in Siena in 2009. And, you could really feel the mood in the meeting kind of went down when they presented these things. And I'll always remember a few months later when Ranga sent an email to Jim and to myself saying, I think we are now ready to do a new study of nitisinone for AKU. And that was because of the work that he'd been doing and that Jim had been doing on the animal model, but also, I believe, on the natural history study. And that's when really we started to launch the idea of a consortium, of the Developer Cure Consortium. And so I'll end with a meeting in Geneva with Tony at the first ever World Orphan Drug Congress, which was in November 2010, where Tony was there for his company PSR, and we started chatting. And Tony said, this sounds like a really, really interesting initiative, and I would love to help. And that was crucial because Tony came also with all the understanding of orphan drug development and the regulatory process. That was absolutely crucial. So yeah, that's just a bit of a kind of, in a nutshell, the first 10 years. I'll hand over to Ranga, who will say more about, fill in the gaps, and also say more about the following 10 years and about AKU. Ranga, over to you.
Thank you, Nick. That's a very good intro. I'll just pick up the baton from there. I think the devastating news was really the fact that the nitisinone trial in the NIH had failed, because we had got going with the intention that nitisinone would be approved and we would start using this drug on our patients in the UK, because we'd done a national lottery funded identification program where 16,000 GPs were surveyed postally, and we came to know of 81 patients by doing that. So starting from 4 patients, we ended up with 81 patients following this identification study that went on between 2007 and 2010, something like that. So this US trial having failed, as Nick has said, we thought it might be because there were too few patients, because they gave 20 patients nitisinone. And just to sort of tell you what nitisinone is, it was first developed by ICI as a herbicide, and purely by chance, I won't go into the really interesting story as to how this happened. They realized that when they were doing toxicology studies on nitisinone, that the rats and the animals that were given nitisinone developed eye problems. And when they tried to understand why these rats developed these eye lesions, they found that this was because it inhibited a pathway in the rat. This is the tyrosine pathway. It inhibited an enzyme in the tyrosine pathway. And this enzyme is called para-hydroxyphenyl pyridinoxidase. Sorry, it's a very long name, but it's easier to call it HPPD. And there was a doctor and a scientist in Sweden who was actually working on this, and he realized that this nitisinone could be used to save lives of a different condition called hereditary tyrosinemia type 1. And this was in 1991. So they tried nitisinone, and it was life-saving in that child. And therefore, since then, it's been used in this other condition as treatment, life-saving treatment. Somebody in the NIH realized that the nitisinone could also work in AKU because of the way, because of its mechanism of action on acting on the HPPD. So this was the trial that failed in the NIH when they tried to follow through on that. So the reason we thought it failed was because it was too few numbers. We thought it wasn't long enough. They only went for 3 years. And we thought but they didn't have the right endpoint because they simply decided after 3 years of study to decide on the success or the failure of the trial by simply looking at one endpoint, which was the rotation of the hip. And we thought those were really important reasons why they failed, because to look at the whole disease and to pick out one feature like that to decide whether the trial was a success or not, we thought it isn't right. This is why we developed the AKU Severity Score Index. Just to give a bit of information about what alkaptonuria is, I know Nick has introduced this already. It's in alkaptonuria, the enzyme that's missing is the one just below HPPD, okay? And this enzyme is called homogentisate dioxygenase. And in people with AKU, this enzyme is lacking. So therefore they can't break down homogenic acid, which then accumulates, it comes out in urine, coloring it black, and it also deposits in connective tissue in the body in a process called ochronosis. Basically looks like melanin in the body tissues going black, but it's more than that, more than just the color. The tissues become brittle and they break down, and that causes the whole disease. And that disease is osteoarthritis, like damage to the spine, to the joints, heart valve involvement, osteoporosis and fractures, ruptures and of the tendons and ligaments, kidney stones. And it's an inherited disease, which means brothers and sisters get it rather than children. It's what's called autosomal recessive condition. The other key feature in terms of our designing all these studies is that this is a very slowly progressive disease. So it only starts with the age of 30, so it has a delayed onset. It progresses very slowly thereafter. And for all those reasons, we have to take this into consideration. Not because it is reversible, and as I've just told you, it's multi-system disease. And not everybody has all of these features, which makes it really tricky to try and come up with an endpoint with the number of patients of this ultra-rare disease that Nick has just said, 1 in 250,000. There are not very many of them around. So to do a study that's going to give us a benefit is quite challenging. So I'm just going to move on the timeline now. To develop nitisinone further, we did two main things in 2011. One was we approached the highly specialized services and got them to commission a service for people with AKU in the United Kingdom. Really, it's not the United Kingdom, it's England and Scotland who really buy into these services. And therefore, we set up the service in 2012, 1st of April. And since then, we've been giving 2 milligrams of nitisinone to patients in England and Scotland. And the great advantage, the reason why we did keen on this is you're not constrained by duration of treatment. So you can study them well beyond 3 years, 5 years. We've now been using nitisinone for 10 years. So we can really see whether nitisinone is making a difference to the disease without any trial constraints. So that's hugely important. And of course, it's benefiting our patients as well. So the second thing we did in 2011 was we applied to the European Commission Framework Program 7, asking them to support a program to develop nitisinone for the purpose of regulatory approval, because we thought it's not just the UK, there's AKU in Europe as well, and we came to know that there are over 450 patients in Europe. So we went down this route of developing nitisinone for EMA approval. And I'm sure you're going to hear more about the details of how we went about it, so I won't go into too much detail. And doing this would really help patients in Europe and in the UK and elsewhere. So the EMA, just to finish off on the timeline, the EMA approved nitisinone for adults in AKU in September 2020. Once DevelopACure was completed, DevelopACure comprised of 3 different studies. One was a dose response study, which you might hear about soon, because we thought the NI trial failed because the dose wasn't right. So we carried out a dose response study. And we also didn't know when the disease actually took hold. So we did a cross-section study called SOPHIA. And then, of course, we did the 4-year randomized clinical trial. And all this was for 6 million given to us by the European Commission. The 4-year clinical trial started in 2014, May, and finished in February 2019. And cutting a long story short, it was successful. The EMA approved nitisinone for adults without capnuria in September 2020. The European Commission has also approved nitisinone for use in European states in November 2020. And now we're working towards access. Not just the job isn't over once it's approved. You now have to make it available at the grassroots level, and therefore we're still working on that. And I'm sure you're going to hear more about how we're doing that. So I will stop there, and I will give this back to you, Rick. So over to you.
Guys, thank you so much. A really amazing and in-depth overview of what you did, and just sets us up perfectly for the conversation. And I've heard this story multiple times, and I still learn 3 or 4 different things I've not even heard before from that introduction. So thank you for that. And I'm sure we'll be getting questions from the audience coming in, no doubt. But what I want to do now is try to pick out some major themes that we can hear from that narrative you talked about. And when we've been talking about this session in preparation, one of those first things to come to is that early stage preclinical work. How did you go about getting prepared to launch this study and to think about working effectively in AKU? And Jim, I think it'd be great to turn to you initially just to get some of your thoughts on that early work in developing the model systems to work in alkaptonuria.
So as Ranga said, the key issue in AKU is an inability to break down this molecule called homogentisic acid, HGA, and that circulates and it deposits as pigment in tissues. And from the American studies, it was quite clear that nitisinone would block the production of HGA. But of course, what they couldn't show in people is that it also prevented this ochronosis. And that's why the mouse model was so useful, because it gave us a very good confirmation that nitisinone not just reduces the amount of HGA, but it completely blocks ochronosis. So we really knew then that this drug would be successful at stopping AKU progression in people. We just had to prove it. Another thing that the basic research helps us with is to develop. So, for example, in the mouse, we had to develop an assay to measure HGA, and of course, that eventually was improved and morphed into the clinical assays for HGA, which was so key in the clinical trials. Also, we needed to find out ways of measuring ochronosis, and in the mouse, the ochronosis is much milder than in humans, because they live for a much shorter period. Basically, their lifespan is a lot shorter. So we have to develop ways of actually seeing ochronosis even at an earlier stage and more sensitively. So I think that was the major contribution of the preclinical research. As I say, it taught us that nitisinone would work, but we still had to prove it. I guess the other interesting thing from the preclinical work is that this wouldn't have happened if we hadn't had Ranga involved, but also the AKU Society, and the patients, because they were the driving force, and it's very difficult to get funding for rare diseases. Some funding authorities say they will fund it, but they will fund rare disease research, but it's very difficult. For example, the major osteoarthritis charities weren't interested. They didn't fund us. We applied many times to them, and they wouldn't fund us. And yet now AKU is the one form of osteoarthritis now that we've got an effective therapy for. So key to this is the patient groups and small amounts of pump priming money. And then Nick was a real driver in us going to the big lottery. again, there we were lucky because we actually got the last big lottery grant that was given to medical research. Because after that, the money went to other projects. I think it was the Olympics, actually. When we went to the EU, I think by then we were more confident because the EU has got an established record in funding rare diseases, and also all of the pieces of the jigsaw had started to fall into place. So Ranga had an effective way of measuring the disease severity. We'd learned from the Americans that we needed this more complicated looking at the disease progression. We knew the drug was safe and would work. I think I'll stop there.
No, thanks, Jim. That's brilliant. I think you just gatewayed into what I want to talk about next, really. And Ranga, Jim's really mentioned there the importance of how you developed the severity score and how using that as a different endpoint. So I wondered if you could just touch on the early work in that autopsy and what that told you about AKU you didn't know, and how that led you to developing this new outcome measure, because this is crucial in everything you did.
Yeah. Nick has already mentioned that Archibald Garrett was studying alkaptonuria in 1902, and he published in the Lancet in 1902. But really, 100 years later, there was an article in the New England Journal of Medicine remembering his contribution, because he was the first to recognize that Mendel's laws of inheritance applied to human disease for the very first time. So, and this 100-year centenary article was all about alkaptonuria and the nitisinone. And I thought I knew a little bit because I'd read this article. And of course, when we did the postmortem on this patient who sadly died in Sleaford and donated a body, I realized when I was looking on at the postmortem done by my colleague, histopathology expert, that I knew nothing about AKU because we didn't understand it. So we were therefore able to give a direction to a research program. We were able to ask real questions as to what are the gaps in our knowledge and what do we need to do to improve the understanding and treatment of this disease. Jim has already mentioned the animal model, and the animal model was really key because there was a sudden doubt following the NIH study having failed, whether nitisinone was actually a good drug for AKU, whether it would actually have any effect on the disease. So the mouse model, Jim, was actually crucial because we were able to show that the nitisinone stopped the octinosis formation in the mouse completely if you started the mouse with nitisinone from birth. And if you gave nitisinone halfway through after the pigment had formed, it prevented any further pigmentation. So it was really, really crucial. And Jim's contribution was really important for it. As I've already told you in my introduction, alkaptonuria is a multisystem disease. And just measuring one feature, like a lateral rotation of the hip, to decide on the success or failure of a treatment is not right. It is illogical with small numbers of patients. So therefore, I came up, I suppose it's quite safe to say because I did think about it in conjunction with the statistics colleague to develop this a composite score. And again, there's a contribution from Professor Tim Cox, who encouraged us to go down this route when we were in Siena hearing about the NIH clinical trial not working, to develop what's called a composite score, bring in all the features of alkaptonuria, try and come up with a way to score these features, and then come up with a score so that you can actually see how much disease a patient has. And then you can follow that patient using the score to see whether that score worsens, stays the same, or gets better. And that was the advantage of having the AKU Severity Score Index. And we could actually develop a clinical trial which for fewer patients by using this score. Again, the AKU Society, as I've already said, have been, I'm part of the AKU Society, by the way. I'm also a co-founder of the AKU Society. I'm also a trustee. I'm also its medical director, and I've known Nick and for very many years. We're colleagues, friends, and we work well together. And of course, the AKU Society has been fantastic in funding all these developments for AKU, because without that, as Jim said, we will be struggling. we wouldn't be able to make the kind of process in such a short period of time if you didn't have the money. I don't think I want to say anything more at this point, Rick. I'll just keep my powder dry.
Django, no, thank you. That's a brilliant touch on that. And I think this severity score measure is crucial, and that development of an endpoint that you can use for your rare condition and the way you deployed it is something that lots of other groups would be interested to find out more about and look into. So thank you for touching on that. But key there is that theme that we've touched on throughout this conference already, was collaboration, right? The fact that you were collaborating, between Jim and yourself, the clinical and the preclinical side, with the AKU Society, and actually bringing in other partners. So it'd be great to hear a bit more about how you approached building that collaboration when you decided you're going to go in to build a consortium for the EU. So Nick, I don't know if you want to take us through some of that journey.
Yeah. I mean, I think, there's a few, I think, really important points about why we succeeded over the years, which we'll go through. And I think collaboration is right up there at the top. I know from my point of view, as I said, I'm not a scientist. Last time I studied any science was when I was at school in France doing my baccalaureate. And so I had very little understanding really of the disease and what was going on. I spent a lot of time Googling, trying to catch up in meetings and understand what was happening. But that's why I think as a patient group, you need to identify, and this is to all the patient groups who are listening, if you haven't already, you need to identify your scientists and clinicians, your Ranga and your Jim, okay, who really will understand all the science and all the clinical stuff around the disease. I can remember the first meetings I used to go to, and I'd be just like, like people would talk about a phenotype, and I remember thinking, what the hell is a phenotype? And be Googling phenotype and trying to understand it and stuff like that. So you need really the key people who really understand this and who are very experienced in what they're doing. And then building a team, I think, around that is absolutely crucial. And finding, basically, my theory has been, and not just in rare diseases, but it was the same in international NGOs, is that most of the problems we face have already been overcome by someone somewhere. Okay. And it's about trying to find them and then putting all that together so that the new problem you face, which no one has overcome, can be overcome. So basically what we realized, we had amazing clinical and scientific knowledge with Ranga and Jim. Okay. What we didn't have was the regulatory knowledge, and that came in with Tony. Okay, Tony Hall at the World Orphan Drug Congress, and he came with all that experience and all that, and then also all the clinical trial kind of management, which came in with Krista, who was working at the time at PSR, who were this fantastic CRO that helped us through it and all that, and then also bringing in the company, Swedish Orphan Biovitrum, was absolutely crucial. You'll have to remember that when so they had been supplying the drug for the trial in the States, and when that trial failed, Sobi, who are a business, basically were just like, this is too complicated. AKU is too difficult. It takes too long, over too long time. We've done what we can. We need to move on. And it took us about a year and a half to convince them. We also met them at the World Orphan Drug Congress, their president and CEO. And what was really good was that they were open to dialogue. Not all pharma companies are open to dialogue. That is really important. And to the pharma executives who are listening, I say, you need to be open to dialogue. And if you're on this virtual conference, it probably means you are open to that. You need to engage with the patient groups. And Sobi were willing to do that, even though they'd really had their fingers burnt. And it took about a year and a half of backwards and forwards. They came to our meeting in Cambridge. We went to see them in Sweden at their headquarters. We had phone calls, teleconferences, and all that kind of stuff. And then eventually, they were just like, actually, you guys are a solid team. You've got a very good scientific and clinical basis. We had done a patient identification campaign, which was Ranga's idea, where we contacted all 50,000 GPs in the UK. We had the animal model, we had the cell model, we had, all that kind of stuff, and an emerging consortium. And they were just like, yes, you guys are serious. Let's give this one last go. And what was even more amazing is that I once did a joint presentation with the former CEO of Sobi, Jeffrey McDonough, at a World Orphan Drug Congress in Geneva a few years ago. And Jeff explained the 101 reasons why Sobi should not have worked with us, which was basically that the patent was going to run out very soon and they would never make their money back. Okay. And he said, fundamentally, they decided to go with us for reasons of values, and also for philanthropic reasons. They were just like, look, if we don't help them, no pharma company will. And so we were really grateful, and their team were fantastic. There was particularly two that spring to mind, Matthias and Birgitta, but there were many others from Sobi. You know, they were absolutely terrific. And so that was really important. And then there was actually setting up the clinical trial centers, you know. So we had in France, the Hôpital Necker Enfants Malades, which was really interested, and they got involved. And then crucially, Slovakia. So whilst in most of the world, it's ultra rare, in Slovakia, it's about 1 in 19,000. And Professor Joseph Revensky, who ran the National Institute of Rheumatic Diseases, and Andrea Zatkova, who ran the AKU mutation database, were absolutely crucial. So it was all about collaboration, and that's what I believe. And without the collaboration, you just won't get there in a rare disease, unless you're a company with hundreds of millions and all that to invest, and which we didn't have. You know, so I'd say that's absolutely crucial.
Thanks, Nick. And Krista, I just want to come to you here. And Nick mentioned the way you came into this. I'm interested in your perspective of coming in and joining this collaboration, this group. What you saw of it, what you saw that was different, and how you were able to work across all these different sites and different interests in your particular role.
Yeah, so it was very interesting. Actually, I was introduced to the whole program through Tony Hall and Nick, and it was immediately obvious that all the consortium partners were really working together as equal partners. What you often see, I've worked on one other European funded study project, where a pharma company was also involved because they were supplying the study drug, just like Sobi were supplying the study drug in the example we're discussing today. But in that case, even though it was officially also a European consortium, it was clear all the time that the pharma company was the boss. They decided. And it was all about also their commercial interests. And what we usually see with in the contract research organization world, so where we are commissioned to run and manage clinical trials, very often trials are centered around commercial interests and shareholders' expectations. While for the Develop a Cure project, it was clear from the start that the center of everything was the patients and trying to get a treatment to these patients that had no treatment at all. And everyone seemed to, have the same interest. And I think that this example of a consortium was the perfect example where everyone's role was clear. Everyone was, part of that consortium for a clear reason. Everyone had their own expertise. And there was loads of mutual respect between the different partners. Everyone respected each other for their expertise and their contribution to the project. And, that's what a consortium should be like. And then, of course, with two very enthusiastic people leading the project, Ranga from the scientific and medical point of view, and Nick at the time from the patient perspective. It was just a really good setup, and everyone was happy to be part of this consortium, and everyone was really dedicated. So it was a unique experience for me to be part of this journey as well.
Thank you, Krista. That's really, really helpful and interesting. And yeah, Ranga, please go. Oh, you're muted right now, sir.
Unmute myself. I completely agree with both Krista and with Nick about all of this. I think we also had a few other partners who did really well. I think, we had Nordic Biosciences who came in actually, thanks to Jim, because he made a contact with them in one of our arthritis conferences, I think. And they provided very good support in terms of biomarkers. And of course, Jordan was very good as well, because we're running into problems with recruitment. And I think they were also very important in terms of partnering and helping with that. I don't know whether Nick wants to mention about the AKU societies elsewhere, but I think we had a very large number of partners in this. I think we had 12 partners who develop a cure.
Yeah, 12. Yeah, I think 13, 12, 13. Yeah, that's right. Yeah, if I could just add to that, Rick, so what we did is helped local patients set up patient groups around Europe and the Middle East. So in France, my parents actually set one up in France, in Italy, in Slovakia was really important, in the Netherlands, Germany, I'm sure I'm missing out some, and also in Jordan, so we had a very enthusiastic doctor in Jordan called Mohammed Al-Zbou, whose relatives were affected by AKU, and actually found out about AKU, I believe, Jim will know the detail, when he was on an exchange with the University of Liverpool, and discovered about AKU there, and then went back and identified many relatives of something like 50 with AKU, which was really quite astonishing, and set up an AKU society, and 19 of them participated in the studies, But also, I think, just to respond to that question that's just come through, how do we patient groups persuade busy doctors and researchers to help us? It's not easy, it is really not easy. It's about taking time, and then it's about slowly building a team, and that's the thing. And then it's also about getting the right funding at the right time. But it is really about the right people, and it's about the right mentality. That's what we found. If you find people with the right mentality, who are not doing that for their careers or anything like that, but are doing that fundamentally because they want to cure a disease which is ignored and abandoned by everybody else, then I think you're onto a winner. So how do you persuade them? Well, I think one of the things, actually, one of the tricks, and I think that we did earlier on, and this was Jim and Ranga's idea, was actually about having annual scientific conferences. And the first one we did, I can't remember if it was 2006 or 2007, there was just like a dozen of us or something. I remember seeing the picture the other day. I think it was at UCL or wherever. There were like a dozen of us, in that conference. And, years later, we had 70, 80 people who would come to these things and all that. You start small, and it's about perseverance, setting the things in place and continuing going and finding the right people who are willing to give you the time.
Thank you, Nick. Krista, do you want to come in?
Yeah, just a short comment. I heard from another rare disease advocate some years ago, and I've always remembered this as a kind of advice for exactly this question: how do you, get through to these doctors and scientists who are very busy? She would start contacting people, and every time she got a no, I'm not interested, don't have the time, whatever, she would say, okay, thank you very much, but please give me 2 other names that I can try. And it's a very simple trick, but it for her, it definitely worked, and definitely perseverance. Yes.
Thanks, Chris. That's a really nice tip. Ranga, we'll go to you, and then we're going to move on to the next theme. Ranga.
Just a quick comment on that, because I am a clinician and I got into AKU, and I was very busy when I first came. So this is quite an important issue. I think you should turn it the other way around. If you're a clinician, you're busy. Therefore, if you work with an AKU society type entity, get them to do a lot of work with you, then you're not doing as much work, and it's good for you, it's good for them. So I think that is important. I think we're all busy, but I think if you come up with ways and build a relationship based on trust and professionalism, I think it's possible. I mean, when I came in, I told Nick and Bob that I haven't got time. So I said, you will need to do a lot of the work, which they have.
You found some time as well, Ranga, which always helps. Thank you, guys. That's really helpful. I'm sure Lucy at Medics Rare Diseases would love to chat to all of you guys in the audience asking about this, and some of the guys here about the different ways to engage medics in the rare disease space. So do check out her exhibition space as well. I want to touch on, Nick, you also called out Tony Hall and the role he played in that early stage in helping to develop the protocol, and really think now a bit more about the development, the design and implementation of the clinical trial itself for a few minutes. So I think what we'll do is just maybe start with Tony about your impressions of that first meeting, and then move through maybe with Krista and Ranga a little bit more about the delivery of the trial itself. So Tony, please go ahead.
Yeah, sure. I mean, maybe I should touch on some of the difficulties that we had in trying to design the clinical program for the development, which I guess became clear fairly quickly. I mean, I guess there were three sort of really main problems in terms of trying to figure out what the development could look like, with the ultimate objective of getting a drug approved at the end of it. The first is that this is a very slowly progressing disease. So in order to run a clinical study with a clinical endpoint, it's quite difficult to design such a study and still be able to confidently have a clinical measure that you can use. I'll come back to that in a minute around the AKU severity score index. The other is that we knew that we wouldn't be able to blind the study, which makes it difficult for several reasons. One is that the regulators don't like unblinded studies for registration, but also because we knew that if we had a long-term study and patients knew that they were not receiving active treatment, that it might be quite difficult to get patients to engage with us for the duration of clinical trial. And I'm still astounded to this day that so many patients stayed in the study for so long. And the reason we couldn't blind the study is because homogenetic acid, it's produced at very high levels in AKU, and most of it's excreted in the urine, and then it oxidizes on standing. So the urine goes dark in color or black, and that means that it's actually quite easy to figure out if you're on the nitazoxanide, because if you're receiving the treatment, then your urine doesn't go black anymore. And the other one was that we knew right from the beginning of applying for the funding was that we were only going to have 6 million euros to play with. And in drug development terms, that's nothing. I mean, that's less than you would often spend on a small proof of concept study. So we had to somehow do a whole development program for this amount of money. So there were quite some challenges really right from the beginning. So the original idea was, well, we've got a great biomarker. We've got homogenetic acid. We know that the levels increase very significantly in AKU, both in the urine, but also in the blood. You can easily measure that. And we know that nitazoxanide reduces the levels. So, and also, as I think Jim mentioned earlier, had also been shown in the animal model that when you block that production of HGA, that you can also prevent your chronosis. So we thought, okay, we'll go to the regulators with the proposition that the development program should be focused around this biomarker endpoint. So we give the drug, we reduce the HGA. Okay, thank you very much. Let's have registration. We kind of knew that they probably wouldn't go for it, and indeed they didn't. And the reason that, or at least they didn't fully. And the reason for that is because even though this is a sort of obvious biomarker, there's still quite some unknowns around it. For one thing, the actual direct link between HGA and the clinical disease in humans is not proven. So this is an unvalidated, let's say unqualified endpoint. And trying to make that bridge using the animal model is quite difficult, because normally you need some clinical human data to be able to show that. So that was already going to be an issue. The other is that you know, even if you accept that reducing HGA is likely to lead to a clinical benefit, the question is how much do you need to reduce it? Is it 90%? Is it 95%? Is it 99%? And of course, the answer to that is really difficult, because you would imagine that the more you reduce it, the better, but probably bearing in mind that this is a slowly progressing disease anyway, you would think that if you reduce it by, say, 90%, that would already slow the disease progression down very significantly, but it's not proven. So we went to the regulators with the proposition that we could use HGA as a surrogate, but we also obviously had the plan that we would present a study with a clinical endpoint, which would be based on the AKU severity score index. During the discussions that we had with them, you know, in the beginning they were quite, they didn't really know the disease very well, and I think one of the reviewers actually commented, well, you know, it's just a pigment, isn't it? Not really understanding the role that it plays in disease. But they did eventually understand that this is important in the disease, and accepted that we would do, first of all, this SONIA-1 study, which was a dose-finding study, as has been mentioned already, and then follow that up with a longer-term study. But crucially, they did agree that we could have homogenetic acid as the primary endpoint after 1 year of treatment, with a clinical endpoint based on the AKU severity score index as a secondary endpoint. So they wanted us to show that we reduced the HTA by 99% and have at least a trend in terms of clinical benefit. And so that was really important, because we thought that in this slowly progressive disease with a composite endpoint like this, all the logistical complications of doing such a study, and a big study for a rare disease, that not having to have a sort of statistically significant clinical endpoint was really important. So we were then able to put together a program that actually made some sense, trying to fit all of this into the 5-year framework of the Develop a Cure funding was always going to be a challenge. I mean, recruiting 140 patients from an ultra rare disease is, and I think we recruited those patients in 9 months. Crystal will probably know the answer to that, but I mean, that's just astounding. And I think we should also talk a little bit later about, you know, how that was even possible for, I think there were only 950 patients known worldwide at the time. So to recruit 140 of them into the clinical trial and for them to stay in the study, I think is just quite unbelievable, really. So yeah, so I mean, we had a big challenge, I think, in trying to figure out what this development program should look like, fitting it into the confines of the program, fitting it into the budget, of course, was also quite tricky. But I think getting the buy-in of the regulators was really key, and of course Sobi was just great. I mean, they played a huge role in making all of that happen, and provided immense support from the regulatory side and clinical side on those discussions with the regulators. So okay, I'll stop now. And maybe
thanks, Tony. That's amazing, and we definitely going to come back to the points around recruitment and retention, which are crucial and really helpful for you to touch on that perspective early on of not just how you tackled the trial and put in the context of funding with the regulation as well. So thank you for that. Ranga, I just want to ask you, how did you feel when you're about to start this? I mean, that must have been a moment, right? You're about to start this trial. Was that an excitement, a trepidation? What things were worrying you at the time before this began? Oh, you're on mute. Sorry, sir.
Yeah, it was both at this time. It's a fantastic opportunity to try and do something for our patients. But at the same time, I was reassured, because one of the great advantages of having had the NAC, the National Cancer Center, going at the same time, was we were doing a lot of the things that we were then going to do in the SONIA-2 already for a few years in the National Center. So we really understood the challenges and the sort of patient journey for the SONIA-2 very well, because we had carried that out in the National Cancer Center, and we knew how to do this very efficiently. And of course, it then became an issue of harmonizing across sites. This is where the PSR did such a fantastic job in coordinating and actually keeping track of how things were going in the various sites through regular monitoring and holding people to account. I mean, so yeah, I mean, that's what I would say.
Thanks, Ranga. And Krista, what was your reflection on beginning to do the work and actually doing the work in this setting and that coordination, and what made it successful from your perspective?
I think a key point was, again, also the clinical sites all being very enthusiastic and very engaged. So that's always a huge advantage when doing clinical trials. But as Ranga said, there was obviously a lot of experience in Liverpool with actually doing all the assessments, which was fantastic because we could leverage that experience, of course. But some of these assessments were not routinely done in the other 2 clinical sites. So as Nick said, we had a site in Paris and one in Slovakia. So we had to do quite a lot of training and harmonization and setting up clear procedures so that everyone was doing the assessments exactly in the same way. So I remember even having done one of the physiotherapists in Liverpool made some videos on how to properly measure some of the movement assessments that we were measuring throughout the study. And of course, this was a 4-year study, so we had to continue making sure that everything was harmonized. And also, since quite a number of assessments, the evaluation of the test results was rather subjective. It wasn't an easy positive or negative, but it was, really a clinician's view. So we decided quite early on that to enhance the quality of the data and having it more harmonized, it would be important to do quite a few of the assessments by a central evaluator. So that was also quite a challenge to get central evaluators to evaluate all the results from 140 patients at different time points throughout the study. So that was from the clinical perspective. And then, of course, because patients were recruited from everywhere, all of Europe, I don't remember off the top of my head how many nationalities we had, but it was a large number. And even from outside of Europe, we had, as Nick mentioned before, a big group of patients recruited from Jordan who were traveling to Slovakia to attend. So we had a large number of patients who were, as we call, doing cross-border enrollment. So they were, traveling across borders to attend the clinical research site. And that, of course, meant a lot of practical logistical challenges in regards to travel, accommodation, but also languages, having all the patient-facing documents in the right language, but also having on-site interpreters to make sure that the communication was done well. And I think for all of those practical and logistical challenges, the AKU Society played a huge role. They basically took care of all of that. And I think that's very impressive because the patient experience was extremely important. And I know we'll touch more on that in a moment with Tony Esmond, but especially since this was a 4-year study where half of the patients was not receiving any treatment, and they knew it because there was no placebo. So we had to keep these patients motivated to stay in the study. And a large part of that was communication, which I'm not sure we'll have time to discuss, but that was very important as well, keeping the patients engaged and motivated and informed, but partly also making sure that all the logistics and that all went smoothly and patients would get reimbursed in a timely manner for any expenses they made. They seem like trivial and small things, but they can make a difference of, whether patients continues this 4-year-long, quite burdensome study or decides after 2 years, forget it, I'm out. So yeah, a lot of factors contributed to that.
Thank you, Krista. That's amazing.
I just make an important point there because Krista has highlighted all the challenges we had in getting the trial right. And the way we did it, I think the single biggest reason why we succeeded was the weekly project board meetings.
Yeah.
So we had a meeting every Thursday, right from the start, right from the kickoff meeting in, I think it was November 2012. And right until February 2019. So we were meeting once a week by telephone. Every partner had to send at least one person to this meeting. And we were able to solve issues. We were able to pull up people who were a little bit slow and support them. And this was really important. I think it's a fantastic model to follow. And I think it's great for a complex project to be delivered.
Thanks for bringing that up, Ranga. That's something I really want to touch on, and I appreciate that hugely. I think it's a hugely important point that just you had that contact, you had that communication, you had that strong leadership as well from yourself to drive it forward. So thank you for that. Kieran, I think this is by far the obvious point to bring you in, and Chris has begun to touch on some of the challenges AKU Society faced and the role you played. I mean, you've got a trial where you've got to recruit 140 patients internationally, excluding a bunch of patients who are already getting the drug in England. You've then got to keep all those patients on the trial when half of them know they're not getting the drug. What did you do? How did you tackle this problem? And what for you were some of the crucial steps at the AKU Society?
Yeah, so it sounds like it's a difficult thing to do, but it's one of the main important things about the AKU story is that the AKU Society was involved right from the start, but not just in the actual clinical trial design and all that kind of scientific and medical stuff, but also making sure that the patients were informed and aware at every point of the journey. So even before we were ready to recruit, we had said from what we learned through the NAC and what we learned through the medical stuff that this drug was their best future for an effective treatment for AKU. So when it came to recruitment, from what I, vaguely remember and what I've talked to my previous colleagues is that actually getting them to say that they'd join it was actually quite easy. And it even got to the point where people were extending our network themselves because they knew in their home country of more patients that we weren't aware of. So the patients became these key partners. And the greatest and most useful tool that we had at our disposal was the hope for a future of our AKU. So as Tony said, and as Krista said, that it's very difficult to look someone in the eye and say that you might not get this drug that could just effectively stop the progression of your disease. But because of the honesty and the work that we did right at the beginning and continue throughout the trial, is that we told them that there's a future generation that may not grow up with the damaging symptoms of the disease. And your sacrifice and the time and all the effort that you put into it personally means that this generation might potentially have the first effective treatment for AKU. And so I'm sure Tony will fill in the blanks there a bit more and actually have that patient viewpoint. But that is such a crucial tool for it. But it's the honesty and the involvement of the AKU Society with the patients themselves that was crucial in recruitment. So as Krista said, we were doing the cross-border recruitment. We just couldn't recruit people from Scotland and England because of the way the NAC was running and the whole thing, the off-label nitisinone. So there was all the challenges that came with that, and I'm sure I don't really have to go into that in too much detail because it is difficult. But again, that hope is universal. I can't say that enough. So when we did recruit, as Krista said, one of the difficulties was the small minor things that you might think would not really be of impact in something like this. But from the Liverpool side, one of the things that we saw mainly in the middle, near the end of the trial, was that people were getting getting fatigued because it was a long process. So we kind of changed our view of it. We kind of changed our approach to it. And part of that was to say to them, look, not only are you sacrificing your time and helping us out with this trial, but we're going to fly you from Paris, Belgium, Italy, all over Europe to come to Liverpool for a week. So why don't you go and relax and in between your things, don't stay at the hospital, come to a hotel that we'll book for you, go see Anfield, Rick, and really try and work out how we can extend this and make it a little break, But also it's the expenses and the flights. So before, that was booked directly by the trial site, which it's fantastic that they could do that, but it didn't really ask the patients what they wanted, So I ended up booking flights directly for the patients, talking to them directly, not through the mirror of the clinical site, but actually doing it directly. So asking what airport they wanted to fly to, what time, if they could do it around work. They had to, instead of flying from like Pisa to Paris to Liverpool, if they're a direct flight, I don't mind staying an extra day, I'll pay for that, etc. Plus then expenses, because by then it had been many, many trips to Liverpool, and unfortunately, just for the admin burden of the NHS and stuff, it was difficult for them to actually get reimbursed. Now, one trip, I suppose, would be fine, but over 3 or 4, it was getting to the point it was actually quite a lot of money. So we just said to them that we will pay it directly, then we can claim your expenses from the actual trial site and the money from the European Commission. So that actually worked, and it did directly stop people leaving the trial, because there is a limit to the milk of human kindness, and if it actually costs money for you to do it, then sometimes people just cannot afford it. Plus, the AKU Society was involved in dissemination. So from the start, we were honest to patients. We told them as much as we legally could. We said to them that this is happening. Ranga's done this. Come see Ranga talk about this. Come see Tony. Come see Jim. Listen to what you're doing. Listen to the wider picture of your time, your sacrifice again. Be part of the wider picture. So we had a developing website that the AKU Society run directly. Through that, we had blogs from individual researchers, also videos and trial updates, and pictures of other patients as much as we could again. Plus, we had our social media. So right at the start, as Ranga said, we had the patient identification campaign. But also through there, we got some money for an information centre at Liverpool before we diversified to Cambridge. But social media, even then, was a really good tool as a way of disseminating what you're doing, but also to talk directly to the patients again, away from that kind of that prism and mirror through the clinical trial. So the AKU Society being involved in it, both in recruitment and retention, was absolutely crucial, because without us, you wouldn't have that direct link to the patients. We heard what they were saying, because this drug is so transformative and it's such a big hope for the AKU patients at the time that they felt sometimes they didn't want to burden with Ranga with these tiny things that was really, really starting to grate and annoy them. So they were just doing it for the benefit of the society, instead of actually saying to, it's actually getting to the point where I need help with this now. So that's where we stepped in. But also, as the voice of the patients, we told them what was going on. And I think all clinical trials should be like this, because it just makes it easier. And, no problem too small. The main thing that I said to patients when they were saying about it, if they don't like the sandwiches at the airport, then I'll see what I can do, But Tony was there. He did it, sorry, Rick, am I still in your job there? But
No, go for it. No, I absolutely. I thank you for that. I think it's really good. And yeah, I was going to say, Tony, I mean, we've left probably one of the most important voices till the end, really. You were there, you're an AKU patient, and you on this trial. I mean, what of this did you see? What was your perception of it? And why did you want to take part and keep taking part in this study?
Well, I think the thing is, it's like when you've got a condition and you know that there's a potential, perhaps treatment for it, it obviously gives you some incentive. And also, Ranga sort of touched on it before when he mentioned United Kingdom, but then went on about England and Scotland, because I come from Wales, obviously part of the United Kingdom, but their health system operated sort of differently. So whereas the English and Scottish patients had access to the National Centre and then had access to the nitisinone, with the Welsh patients, we didn't get that. And in fact, we had to sort of fight really to be able to get to the National Centre. And we only managed to get to the National Centre on the condition that we weren't actually given the nitisinone. So we never got the nitisinone, but we could go to the National Centre. And the reason they wouldn't was because obviously it wasn't licensed. And as far as they were concerned, that was it. So we sort of attended at the National Centre. At the time, there were only 4 of us Welsh patients, but that gradually increased. And then obviously we became aware of the clinical trial that was going to come along. And obviously Ranga spoke to us all about it, because obviously it gave us possibly the opportunity to go on the treatment arm and to actually get nitisinone. And also back in Wales, we're also monitored as well with the consultant team here as well. And they also sort of advised us that perhaps it would be a good thing to sort of put ourselves forward. I know a few of us who sort of used to communicate with one another fairly regularly talked about it. And in the end, 2 of us decided that, yes, we would put ourselves forward, but only after discussing it through what it actually entailed, because I suppose one of the biggest concerns was we'd managed to get to the National Centre. We'd seen what it was like to be able to have all the tests done, to get the reports, to know all of a sudden what you needed perhaps to work on. And my GP always said he always used to wait for that report because it gave him something to go on. And what we didn't want to do in some ways was to jeopardize that, but we knew it was going to be different. But it was also that we knew that obviously if something showed up, if we went on the clinical trial, that we needed sort of to be seen by a consultant back in Wales, that would be highlighted with us. So that gave us some reassurance as well. So two of us decided to go on it, and two of us were quite close and lived really close to each other as well. And we both went up to Liverpool to start going on to the trial and to find out whether we get onto the treatment arm or be randomized for the non-treatment. And as it happened, both of us got randomized to the non-treatment arm, which Ranga was not very happy about, but that's the way that things go. And we had two quite opposite reactions from it, because the other person was quite upset that they weren't going to be able to get the nitisinone. Whereas I was sort of saying to them, look, it doesn't matter, you know, we're part of that trial. And at the end of the day, most probably we got more of an important part to play, because without them having the baseline of us to follow and see how the condition was progressing compared to the ones on the nitisinone, that the study wouldn't work. So with the other person, we sort of used to link up quite regularly as well and talk about things to make sure that how things were going. But the biggest thing for me was, and the biggest motivation really wasn't about me. My biggest motivation was always been about people who are younger than me, people who perhaps haven't been diagnosed with the condition, and knowing what people were potentially having to go through. I know that possibly if there was something there that could slow it or even stop it, was something that you had to work for. And 4 years, when you say 4 years, sounds a long time, but those 4 years actually went quite quickly. We knew exactly what was happening. We knew when we were having to go to the centres, when we were having tests, what tests we were having done. We were kept informed all the way. And a nice part for me was when I was going up there, I didn't actually attend with the other person from Wales, but I attended with a gentleman from Spain who was always on at the same time as me. And this particular gentleman was actually on the treatment arm. So I could see how he was progressing, receiving the nitisinone. And it was quite nice of how a sort of relationship developed between him, and his daughter used to come very often with him as well, and how she could tell me about what her dad was like. And her remark to me in the very first meeting when we were talking was, she said to me, I lost my dad. He's not the dad that he used to be. And, you know, you could see by him how he was holding himself, you know, the way his hands were with his fingers, his walking, how he couldn't go upstairs very well. And you could see all of that right from the very beginning. And then as we started going through the years, you could gradually see a difference with him. And you could see how his face started sort of loosening up, really, and how he started smiling, how he could open his fingers, how he started walking much better. But the one thing was, he felt really guilty. And the reason he felt really guilty was because I wasn't having the drug. And I had to keep reassuring him and saying to him, it's not a problem. You know, we're doing this for the same things. We're doing it for the same reasons. But seeing him and how it affected him sort of made you want to carry on. Even if you started having doubts, you still wanted to carry on. And the thing about with all the testing and the reassurance that you were given, that was all upheld all the way through the study. So that when you had tests done, if something did show up, you knew that a letter would go to your GP, or you were given something to go away to take to your GP to sort of say, look, this has shown up, we need to investigate this further. So it was all open and above board, and you knew what was actually happening. You knew who to go to and everything. So we had I suppose because we had a real incentive to do it, I think that's most probably why everyone persevered. And like you said, we didn't have to guess whether we were having a drug or not, because we knew, there was no getting away from it. You were either on it or you weren't. So you knew what you were getting yourself in for, really. But I think everybody at the end of the day had that same end goal. What they wanted was to be able to get the drug license so that everybody could sort of benefit from it.
Thank you, Tony. I mean, brilliantly put, and really highlighting the personal motivations, but also the impact of the community more widely there, and the connection you felt to the organisation, but also the clinicians that you're working with too. I think that's brilliant, and I'm seeing a few reactions as well on the platform to what you said. So thank you for that. Time is flying away from us, and I've got questions I want to get to. So what I'd like to do is, Ranga, at this point where Tony got to, it feels great if you could just briefly, in a nutshell, summarize what were the actual results. You did all this work, you got to the end of the trial. Was it what you expected? And you're muted right now.
Sorry.
Don't worry.
I think the results were better than we expected. I think the whole trial was a success before we even started. As Tony said, I think the European Medicines Agency told us that we only needed to show decrease in urine homogenous acid that remained low at 1 year, and we showed that it remained low for 4 years. So we knew right from the start that this was a trial that was going to be successful. The tricky bit was to show some clinical benefit. And of course, we showed that as well. We showed a lot of improvements in pain, in range of motion. We could see the pigment clearing up. We could see improvements in bone density. Although not statistically significant, we saw a big difference in ruptures between those who were treated and those who were not treated. Big difference in fractures between those who got the drug and not got the drug. So I think it was more than we expected. And of course, the AK-70 score index was statistically significant at the end of 4 years. So the metabolic endpoint and the clinical endpoints all were successful in the trial. And yes, I mean, that's a summary of the trial, as it were. Is there anything else you want me to say? I mean, I'm really proud to have been a part of that. Yeah.
That's what I was going to ask. I mean, what was your personal reaction to that, Ranga? Because, this is a story about a huge range of people, but each individual must have had a different reaction to that. And I think you were so close to it. How did you feel at that point?
I felt fantastic because this is such a complicated study with so many partners, so many patients to support, such a long journey for patients each time they were there from Monday till Friday before they went away. And these were, yearly visits for 4 years. I think everybody stuck it out. And I think I'm really proud of the whole team. I think we really delivered for the patients. That's what I think. I think this is all, to improve the health of people with AKU and the prospect of people with AKU.
Thank you. And then I'd just like to ask broadly the team, probably maybe start with Nick and bring Tony and Ranga in a touch. After you got that trial, the aim is then to get the access to the patients, right? And what was the process? How did you find it? And one of the questions from the audience is actually, what do you wish you'd done to prepare for those reimbursement and HTA discussions, in hindsight now, before you got there?
Sure. I mean, I've seen some of the questions, such as were we able to involve this in the discussion with Soviet price and reimbursement and all that. There was someone who became a good friend called Josie Godfrey, who used to work at NICE and is now a consultant. And every time I met her at a conference, she'd say, Nick, are you thinking about what happens after marketing authorization? Because we had this kind of marketing authorization, you know, la, la, la. And it was all about what happens afterwards. And we did have discussions with Sobi. Now, in a way, we've been really fortunate because for the pricing, Sobi did not change the price. One of the reasons is nitisinone came off patent in Europe in 2017, so there was no patent, and they went for what's called a label extension. So it was already a drug that had been approved for hereditary tyrosinemia type 1, and they went for an extension of that label to AKU, so they did not get the 10 years of market exclusivity. At the same time, there were loads of generics coming on the market, so the price of nitisinone has actually halved. You know, overall, you know, it hasn't gone up, it's actually half. So we've been really fortunate with that. You know, that's the first thing. It's not been easy to go through all these HTA things, you know. So Sobi is dealing with the HTAs across Europe, well, everywhere really. But we had some difficulties with NICE. Because basically, you know, the Sobi was not going to make that. You have to spend hundreds of thousands of pounds to go through these HTA processes. You have to pay the fees, you have to do the health economic analysis and all that. And Sobi were realizing they were not going to make their money back in the UK for actually going through this. So they were like, look, you know, we really can't go through this. And at the beginning, NICE were like, well, if you don't go through it, we'll have to give a negative appraisal. And we were like, oh my God, they give a negative appraisal. Will that affect the National AKU Centre and all that? And actually we had some really good workshops and meetings with NICE, and they were absolutely fantastic. And in the end they said, okay, we won't go through the appraisal, and what will happen is that the current commissioning will continue, basically, because also the EMA approval, because it happened before the end of 2020, was also for the UK, you know. So that went ahead, so that was absolutely fine. Now we have an issue with Wales, you know, as Tony Esmond said, we've been struggling to get patients in Wales on nitisinone for the past decade, you know, and we've been tearing our hair out, to be honest. It has not been easy. And to be perfectly honest, I haven't been very impressed, you know, by the Welsh health authorities dealing with this. However, now things are changing, and they are doing an appraisal of nitisinone. So we're going through that process. We're putting everything we can to show that this is just a no-brainer. You know, we did calculations 10 years ago that showed that an untreated AKU patient would cost the NHS on average £100,000 a year. You know, nitisinone is a fraction of that price. It's an absolute no-brainer. All the clinical studies now that we've done show that it works. You know, it's unacceptable if Welsh patients continue not to have access to it. So basically, we're going to keep on fighting. You know, we will always fight for our patients wherever they are so they can get access to this drug.
Thank you, Nick. Ranga, anything you want to add on that kind of access story? Not
really. I know I'm not on mute. Yeah. I know that Sobi are working very hard. There is a mechanism in Europe called MOCA. I'm trying to remember what MOCA is called. I think it's a mechanism of coordinated access to medications. That's what it is. And it's formed of all the countries in Europe coming together to a round table, and Sobi are working with them so that there's a statutory agreement to fund nitisinone locally in the countries. And that's very important because as Nick has just said about Wales and the difficulty of getting the funding for it, while you have competing health agendas like heart disease or cancer or whatever, fighting for this is important, and therefore, you know, that's happening at the moment. Other than that, I don't think there's anything very much to say. I think in the UK, for us, the NICE process isn't happening. So we're going to continue on the National Alcapra Centre, and we're going to continue providing nitisinone there. And of course, we are now addressing what do we do about the 2 milligrams of nitisinone we were giving to our patients with AKU in the National Centre. Now that 10 milligrams has been approved in Europe, we don't want a situation where Europe has access to 10 and the NSE has access only to 2, or patients in England and Scotland are only having access to 2 milligrams. We don't want that. So we are working with the highly specialized services, our funders, to try and resolve that.
Thank you, Ranga. That's fantastic. And to actually be at a point where you've got that access secured through the traditional route is great. And hopefully we'll soon see something in Wales. Tony E, we've got a quick question for you particularly, and people asking, what is it like emotionally and mentally being part of a clinical trial? And did you feel prepared for it? And what support systems really kind of spoke to you? And I'll have to press you for a relatively short answer, but I'd love to get your thoughts.
I think because we were well prepared beforehand, I can't say it really affected me emotionally in any sort of way. Because you knew what was happening, because things were in place, because you knew you had people to access if you had any queries. And I think there was just a lot of support within the patient groups themselves as well. So you knew that if there was a problem, it got picked up on anyway. And as part of your visits, on the yearly visits, they were issues that were actually looked at as well anyway. So you had plenty of opportunity to go through things like that.
That's fantastic. Thanks, Tony, and good to hear. We're coming towards the end of the session now, so I'm going to ambitiously see if each of our panelists could maybe give just a couple of words and your thoughts on maybe the element of your involvement in this study or the study itself that you're most proud of. What stands out for you as a thing that you think is a huge success that you're really proud to see you've achieved? And I'm going to randomly victimize Tony Hall to start with.
Well, one thing is quite difficult. I mean, I think, you know, for me, I'm like everybody else, you know, I'm obviously immensely proud and honored to have been a part of the whole program. I think it's, you know, it was just great that we were able to do such a successful thing with this funding from the European Commission. I guess, you know, for me, I was lucky enough to be, so I had involvement kind of with two hats because I was one of the founders and owners of PSR, as well as at the time also being running my own independent consultancy. So I was able to get involved, like both from the clinical operations side, but also from the medical side and regulatory side, putting together the development program. So that was great to be able to do that. And so, okay, I'll be quiet and let somebody else have a word.
Short and sweet, Kieran.
Yeah, so I just want to say that just thank you to all the patients involved that actually gave their time up for Develop a Cure. But there's nothing better than meeting someone in person, both during the clinical trial and at the end, and knowing that the tiny bit that you've helped towards that trial means that they're now on nitisinone. I mean, you can't get much better than that. That's why a lot of people get out of bed in the morning, you know.
Absolutely. Thank you, Kieran. Jim, any thoughts?
Yeah, I think in most scientific research, you don't really see the end point. You see, you know, for example, I've worked on osteoporosis and osteoarthritis, and we're adding very, very tiny bits to the jigsaw. But in AKU, we were able to add a big bit and complete the jigsaw. Well, complete it to a point, you know, to an effective therapy point. Although there are other questions to be answered. So yeah, so it's actually it was a privilege.
Thank you, Jim. That's a brilliant summary and a really, really strong point. Thank you for that. Krista.
Yeah, so from a clinical operations point of view, I would say the fact that all collaboratively we were able to enroll 140 patients with an ultra rare disease in 9 months' time, and also to hang on to those patients, most of them, large majority of them, for 4 years, I think has been really impressive. But in the end, of course, the fact that the trials were successful, the drug actually is now available to the patients is exactly what Jim has said, the best outcome.
Thank you, Krista. Tony Esmond.
I think for me, it's just really for the whole group of patients to actually see it right through to the end, and to do something to perhaps improve the quality of life for those younger people coming through.
Thank you, Tony. I think it's a message that's resonating with a lot of people in the chat, and really appreciate your thoughts there. Ranga?
I have been saying an awful lot, so I'll keep this short and sweet. For me, what struck me was how much you can achieve if you have a group of people coming together working together, all pulling together and working towards the same end. I think that's what's been absolutely amazing for me in this, and I'm really delighted.
Thank you, Ranga. Beautifully put. And Nick, if you have a thought on what your proudest moment is, and then if you want to sum up how you see this to end the session, we'd really appreciate that.
Well, for me, the most amazing is my boys now have a treatment. You know, I mean, I'm just eternally grateful to Ranga, to Jim, to everyone on this panel, you know, and to all the patients and all that, because it's transformed their lives. They're both on the drug, you know, and it's going really well, you know. So for me, it's just, I'm just absolutely amazed and eternally grateful. I think just to round up, I think the key message, there's two key messages I want to bring across really to any of the patient groups who are listening. One is collaboration. We've talked about that. The second one is perseverance. 10 years ago in 2011, in May, I was at the conference of EURORDIS, the European Organization of Rare Disorders in Amsterdam. EURORDIS have been fantastic. If you're not involved with them, get involved, they're brilliant. And the vice president, I remember speaking to her and she said, look, you know, I told her what we want to do. And she said, look, the battle never ends. You know, it never ends. You have to always persevere, whatever happens. You get marketing authorization, it's then reimbursement. You get reimbursement, you have to make sure the reimbursement continues. You have to, it just never ends. And I think that is what I really want to say to all the patient groups and all that. Just keep going, keep going, keep going. Find the right people along the way, bring them in, work collaboratively, and just keep going. Just never take no for an answer. I think that's the next thing. The third thing is also for us now, we're focusing on other therapies. We're focusing on gene therapy. We're focusing on therapies to deal with the side effects of nitazoxanide. So the research never stops, you know, and you have to continue for that. And the most difficult, I think, is the fundraising, you know, really tough at the moment, but also then the implementation. And then finally, I think something that I'm really grateful for is that, at least for me and for Tony, Find a Cure came out of this, you know. So when we set up Find a Cure in 2012, it was because we were being contacted regularly by people setting up patient groups saying, what do I do? How do I do it? And all that. And that's why Find a Cure has been so focused on supporting these emerging kitchen table patient groups, because we don't want to reinvent the wheel. You know, had we had a Find a Cure around 20 years ago, we might have actually gone faster through the whole process, because people have said, have you thought of that? Have you thought of that? There's money there, there's that, and the other and all that. And that's why Find a Cure is just doing such an amazing job, and also drug repurposing, you know. So yeah, I wanted to thank the Find a Cure team for all that you've done. It's been a really good 2 days. And I think there's great plans for the future.
Thanks, Nick. We've snuck over time, but I think you'll all agree it's been worth it. It's been brilliant to hear you guys tell your story. I'm glad that I managed to navigate the majority of the points, and thank you all for sharing your opinions, and thank you to the audience for the questions. I think we've addressed most things, and where we haven't, we're going to try and get to you guys soon. For now, I should wrap the session up. Thank everyone here, and please come and join us for our final big mainstay session in about half an hour's time, where we'll look at far more examples from the world of repurposing. Thank you all.
Thank you.
Thank you. Bye-bye.